Okay, ladies and gents, this is Chapter 5 from Campbell's Cell Regulation. It's part of our College Board Unit 3 and 4. We started in on the transport proteins last class. Today we have to finish up the other transport proteins, and we'll get going on that. Other transport proteins called carrier proteins bind to molecules and change shape to shuttle them across the membrane. We're going to see a couple instances, a couple examples of this as we move through. A transport protein is specific for the substance it moves, not unlike the enzyme substrate complex that we talked about back when we studied enzymes. Passive transport is diffusion of a substance across a membrane with no energy investment. And I apologize if we've already covered this last class, but if we have to back up a little bit, that's okay. All right, diffusion is kind of the poster child for passive transport. All right, passive. In this case, diffusion is the tendency for molecules to spread out evenly into available space. Although each molecule moves randomly, diffusion of a population of molecules may be directional. What do I mean by that? Well, if we have a bottle of perfume here, and I don't know, I didn't mean to put that in there. Let's see if I can get that out of there. Nope. Anyhow, if we have a bottle of perfume here, we open that bottle, all right, the enzyme, the molecules are going to be in the highest concentration right above this bottle. And if we wait a little while, the molecules will continue to diffuse, all right, and they might make it out to this ugly person's nose over here. And of course, they would continue to move until the concentration is the same throughout the room. All right, we have molecules moving out away from this highest concentration to lowest concentration. That goes without saying. You all know this. All right, at dynamic equilibrium, as many molecules cross the membrane in one direction as move in the opposite direction. And we use a symbol for this, that double-headed arrow, the same symbol that you would use in an equation where you have the equilibrium constant represented. All right, in this particular set of drawings here, this is the diffusion of solutes across a synthetic membrane. Now the solutes are moving this time. In this case, we have molecules of dye here represented by these tan-colored or gold-colored spheres, right? And we have water on the other side of the synthetic membrane, and water is obviously going to have a net diffusion across that membrane in this direction. All right, and they're going to continue to move until you have the same number of, these are molecules of dye, until you have the same number of molecules of dye on both sides of that membrane. Now down here we have diffusion of two solutes, okay, and we have diffusion of the purple going in one direction and the diffusion of the gold going in the other direction. And once again, in the end, you will have an equal concentration on both sides of the membrane of the sum of those solutes, one on the other, as one compared to the other. So you have a dynamic equilibrium once again, but in this case, it's with two solutes. Now, I don't know if my face was right on top of that, but here we have, again, both solutes coming into dynamic equilibrium. So passive transport, again, is the diffusion of a substance across a membrane with no energy investment. Substances diffuse down their concentration gradient from where it is more concentrated to where it is less concentrated. No work must be done to move substances down the concentration gradient. The diffusion of a substance across a biological membrane is passive transport because no energy is expended by the cell to make it happen. Again, that's the term passive here. Osmosis is the diffusion of free water across a cell membrane. Water diffuses across a membrane from the region of lower solute concentration to the region of higher solute concentration. I see I have the word concentration written twice here, until the solute concentration is equal on both sides. So water is moving in response to the solute concentration. Okay, so here we have a synthetic system, if you will. We have a barrier here representing a membrane. We have a higher solute concentration on one side of the barrier as compared to the other. Now, in this case, we have sugar, and sugar in this case is unable to move across this synthetic barrier. So what moves? Well, water moves in an attempt to reach dynamic equilibrium. So water is going to move, as this arrow indicates, from the lowest solute concentration to the highest solute concentration. So water continues to pile up over here until the solute concentration is the same on both sides of that membrane. And you literally have water piling up in this U-shaped tube. All right, so again, just to identify the parts of the system here, we have selectively permeable membrane. Water molecules can pass through the pores, but sugar molecules cannot. All right, and over here you see water molecules cluster around the sugar molecules. This side over here has more solute molecules and fewer free water molecules. So water, this side over here, has fewer solute molecules and more free water molecules. So the water is going to move, obviously, in this direction. All right, water balance of cells without cell walls. Okay, I've noticed that there's a mistake in this. The words higher solute concentration should not be there. So you can scratch these three words out. Okay, that's not supposed to be there. And if I could pause this for a moment. Okay, it should say tenacity. Tenacity is the ability of a surrounding solution to cause a cell to gain or lose water. And we know that an isotonic solution is a solute concentration is the same inside the cell as it is outside the cell. So there's no net movement of water. A hypertonic solution, remember hyper means higher, too much energy, if you will, a person who is hyper. All right, so the solute concentration is greater than inside the cell loses water. And if it's a hypotonic solution, like distilled water, all right, the solute concentration is less than that inside the cell, so the cell gains water. Okay, you may have a mistake on your on this slide here. All right, if we place a red blood cell in a hypotonic solution like distilled water, you could label this distilled water here. All right, the solute concentration is highest on the inside of the cell, so water rushes from where it is highest concentration to lowest concentration, or from where the solute concentration is from low, right, to where it is high. So it's going to rush into this cell in response to the high solute concentration. Or you could say in response to the high water concentration. Water moves from where it's high to where it's low. And in this case, we have a normal cell, all right, and we have I should say a normal solution, and that's what we would say. It's normal or isotonic. So if you were given a saline solution in the hospital, you would have the same concentration of solute in that bag of solution as you would have in your red blood cells. And this would cause no net gain or loss of water. And then, of course, if you had a solution that is hypertonic, and make sure that this one says hypotonic and this one says hypertonic. So if we placed cells in a salt solution, for example, water would rush out of the cells in an attempt to reach equilibrium. And of course, that could never happen, and the cell shrivels up. All right, this is plasmolysis. This is cytolysis. Cytolysis, and this is plasmolysis. Hypertonic or hypotonic environments create osmotic problems for organisms. Osmoregulation, the control of solute concentrations and water balance, is a necessary adaptation for life in such environments. The protist Paramecium caudatum, which is a hypertonic to its pond. Let's think about that. It's hypertonic to its pond. In other words, it has a high concentration of solutes inside that cell membrane. So it's hypertonic to the pond environment. So water is going to want to tend to rush into the cell. And they have a special, oh, it's missing an E on the end of vacuole here. They have a special organelle called a contractile vacuole that can pump excess water out of the cell. So if we look right here and we see a paramecium, you can see the cilia on the margin of the cell membrane here. This is most likely, it doesn't say that it's with it's called it's a Zeiss photo microscope. Okay, so you can see the contractile vacuole right here. This organelle is filling up with water, and then it contracts and it pushes the water out of the cell. Again, fills up with water, and it contracts and pushes water out of the cell. And once again, here's the Paramecium caudatum, all right, with its contractile vacuole. Really cool looking star-shaped structure here. So if this guy's 50 microns, he's about 5 times that, 250 to 300 microns total in length. Pretty big cell. All right, water balance of cells with walls. Cell walls help maintain water balance. A plant cell in a hypotonic solution swells until the wall opposes uptake. The cell is now said to be turgid or very firm. If a plant cell and its surroundings are isotonic, there is no net movement of water into the cell. The cell becomes flaccid or limp, and the plant may wilt. Hypertonic solution, plant cell loses water. Of course, if we place a celery stick, for example, in a salt solution, all right, the water rushes out of the cells in an attempt to reach equilibrium, and of course it would never reach equilibrium. All right, and if this happened to a plant in a live plant, this obviously could cause plant death. And again, that would be plasmolysis. All right, I think we mentioned that in a previous slide. In the red blood cell, where it implodes is plasmolysis, and where it undergoes cytolysis, that's where it explodes.